Cellular Respiration, Part 1, for both Grade 12 and remedial students by Dr. Alamayo. In this lesson, we will see about ATP, coupled reactions, sodium-potassium pump, and the first stage of cellular respiration, glycolysis. If you find this lesson helpful, please subscribe, like, and share to help more students learn. Let's get started.
Cellular respiration is the process by which cells produce energy from glucose in the form of energy storing compound, which is ATP. This ATP is called adenosine triphosphate. Here, ATP often referred to as the energy currency of the cell. That means the cell can use its ATP as the source of energy. Here, if you look at this diagram, this is the structure of ATP or adenosine triphosphate. Adenosine triphosphate or ATP has 3 phosphate group, and also the adenosine triphosphate has the pentose sugar, which is called the ribose. And also in adenosine triphosphate, it contains a nitrogen base. This nitrogen base is called adenine. Therefore, it is called as adenosine triphosphates. Here, the energy released by hydrolysis or breakdown of ATP is used to power many energy requiring cellular reactions. In our cell, different reaction takes place, but this reaction to be occurred or to take place, our cell need energy. Therefore, our cell use this energy to do different activities. Therefore, ATP is made unstable by 3 adjacent negative charges in the phosphate tail. Look, this is the structure of ATP. Here, the phosphate group of ATP has a negatively charged oxygen. These 3 negatively charged oxygen make ATP, which is unstable. Therefore, bonds between the phosphate groups which attach these phosphates called the phosphoanhydride bond. This phosphoanhydride bond is attach the phosphate group that make up the adenosine. Therefore, the phosphate anhydride bond is high energy bond. High energy bond. Here, hydrolysis of ATP. Why are the phosphoanhydride bonds considered as high energy? This is because it has an appreciable amount of energy is released when one of these bonds is broken in hydrolysis reaction. Also, by hydrolysis, ATP can be hydrolyzed to ADP, that means adenosine diphosphate, plus inorganic phosphate plus energy. Then energy is released during this hydrolysis of ATP. The hydrolysis of ATP can be reversible. Here, ATP can be hydrolyzed to ADP, then to AMP, means adenosine monophosphate. But this process can be reversed. This process can be reversible. That is why it is so called as reversible. Then it also regenerates the ATP from ADP and inorganic phosphate by the process of reversible reactions. Here, the coupled reaction. A coupled reaction will involve ATP molecules. Here, a coupled reaction is carried out when 2 reactions occur nearly simultaneously. The first reaction must be exothermic, and that gives off energy, and the second reaction is the endothermic, which immediately uses the energy produced from the first reaction, which means from the exothermic reactions. Now, if you see this diagram or the ATP cycle, here in the hydrolysis, they release out energy release, which is exergenic. But this, in the synthesis of ATP, there is input of energy, which is endergenic. This exergenic and endergenic simultaneously takes place, or nearly simultaneously takes place. If there is such kind of reaction, we can say that it is a coupled reactions. Now, if you take this one, when ATP hydrolyzed, it can be converted into ADP and inorganic phosphate. Now, in the second reactions, when ATP is synthesized, this ADP can be used as an intermediate, and it produces ATP. This is how the two reactions, or the hydrolysis of ATP and the synthesis of ATP, takes nearly simultaneously. This process of reaction can be called as the coupled reactions. Reaction coupling. How is energy released by ATP hydrolysis used to power other reaction in a cell? Here, in most cases, cells use a strategy called reaction coupling, in which an energetically favorable reaction like ATP hydrolysis is directly linked with energetically unfavorable reaction, which is endergenic processes, which needs energy for the reaction process, and the link often happens through the shared intermediates. In the coupled reaction, there should be a shared intermediates. Therefore, meaning that the product of one reaction is picked up and used as the reactant for the second reactions in the coupled reactions. Here, ATP in reaction coupling. Now, here if you see the ATP reaction coupling, when reaction coupling involves ATP, the shared intermediate is often the phosphorylated one, because phosphates from the ATP can be attached with another compound, and the form is the phosphorylated molecules. An example, look at the formation of sucrose, or a table sugar from the glucose and the fructose. Here, this one is a sucrose. Let me see how this sucrose is formed by coupled reactions. In the first case, glucose is one of the monomer of carbohydrate, which is a single unit, or it is a monosaccharide. When it reacts with ATP, now the ATP loses one of its phosphate, and the phosphate attached to this glucose, and the form is glucose phosphate, which is phosphorylated compound. Now, this phosphorylated compound become used as an intermediate for the secondary actions. Look, this one is the phosphorylated glucose. And the secondary action, it can serve as the reactant and react with another monosaccharide, which is fructose. Finally, they produce a disaccharide, which is sucrose. This is how coupling reaction can take place. Therefore, in this case, ATP hydrolysis can be coupled to be for the biosynthetic reactions by this process. Here, ATP hydrolysis can also be coupled to other classes of cellular reactions, such as the shape change of proteins that transport these other molecules in and out of the cell. Example of coupled reaction is that the sodium-potassium pump. Here, this is energetically unfavorable to move sodium ion out of the cell and potassium into the cell. Therefore, because of this movement, it is against the concentration gradient, it is difficult to move or pump sodium out of the cell and the potassium into the cell. But ATP provides energy for the transport of sodium and the potassium by the way of membrane embedded proteins, which is so-called sodium-potassium pump. Now, ATP hydrolysis is coupled to work requiring or energetically unfavorable process through the formation of unstable or phosphorylated intermediate. This along the process takes place in a series of steps that are each energetically favorable reactions. Now let me see about the sodium-potassium pump. Here, normally, the potassium ion is found in the extracellular fluids. That means the fluids around the cell. And sodium ion is found inside the cell. That means in the cytoplasm of the cell. Now, for the normal nerve impulse transmission, the sodium ion has to be pumped out from the cell, and the potassium ion has to be pumped into the cell. But this process which is energetically unfavorable. Therefore, by the process of sodium-potassium pump, the membrane embedded protein, look, this yellow one, which is a protein embedded in the cell membrane of the cell. This membrane embedded protein, when it attaches with ATP, it is closed from the outside directions of the membrane. But when ATP is hydrolyzed, this potassium becomes attached to this membrane embedded protein. Then from the outer side parts of this protein, it becomes open. But in turn, from the inside part, it becomes closed. Then at the third stage, when the ATP reattached to the inside parts of the membrane embedded protein, from the inside side direction, it becomes open. Then from the outside direction, it will close. Now, let us see how the movement of this ion can takes place. For example, in the first case, the sodium ion pumped to be leave the cell, but it cannot pass this point because it is closed. Then, when ATP hydrolyzed, one of this phosphate become attached to membrane embedded protein. Then from the outside direction of this protein become open. As the result, the sodium ion become pumped out, but the potassium ion starts to pump it in. Look, the process. Now, the sodium is moved out, but the potassium is trying to enter into the cell, but it cannot pass this closed portion. Then at the third step, now when ATP becomes again attached to the internal sides or parts of membrane embedded protein, it becomes open from the internal side. Then the potassium totally pumped into the cell. Look, the process. This is how sodium-potassium pump can take place by the process of coupled reactions. Now, let me come to another point, which is the site of cellular respiration. The site of cellular respiration. What is the purpose of cellular respiration? Why cells undergo respiration? It has many purposes. Some of the importance or the purpose of cellular respiration is that here cellular respiration is used to generate a usable ATP energy in order to support many other reactions in our body. In our body, many reactions takes place. But to support the process of reactions that takes place in our body, cellular respiration is very important to generate the usable ATP energy, which is energy currency for the cell. Our cell use this ATP to undergo different reactions in our body. Here, cellular respiration is carried out both by prokaryotes as well as the eukaryote cells. In prokaryote cells, it is carried out in the cytoplasm and the cell membrane of the prokaryotic cells. Whereas in eukaryotic cells, it begins in the cytosol, that means fluid inside the cytoplasm, and carried out in mitochondria. You know that prokaryotic cells has no mitochondria. Therefore, or the cellular process, cellular respirations in prokaryotes takes place in the cytoplasm as well in the cell membrane. Here, if you see the sum up of the reactions of cellular respiration, to completely oxidize 1 molecule of glucose, we need 6 molecules of oxygen. Finally, 6 molecules of carbon dioxide produced and 6 molecules of water again produced, but this energies again ATP can be produced. The main target of cellular respiration is to produce this ATP, but this ATP productions in the case of cellular respiration has different steps, and it can be takes place at different sites of cellular respiration. The first stage of cellular respiration is so-called, it is glycolysis. Glycolysis. Or this glycolysis is called, or it is another name is so-called, the Embeden-Maherov pathway. It is so-called as the Embeden-Maherov pathway. Here, glycolysis occurs in the cytosol of the cell. It does not require oxygen. In the process of cellular respiration, oxygen is important for aerobic organisms, but in the first stage of cellular respiration, that means in the glycolysis, oxygen is not important. This first stage does not use the oxygen for the process of cellular respiration, so that it is the common pathway for both aerobic and anaerobic one. Both anaerobic and aerobic organisms can use the first stage of cellular respiration, which is glycolysis. Therefore, glycolysis is common stage for both anaerobic and aerobic respiration. Here, glycolysis begins cellular respiration by breaking down into 2 molecules of 3-carbon compound, which is so-called pyruvate. Here, this is to show that the cellular structure of eukaryotic cells. You know, eukaryotic cell has 2 main parts, which is the cytosol, which is a fluid-filled portion, and this also it is filled by different membrane-bounded organelles. The first stage of cellular respiration, which is called glycolysis, starts in this portion of the cell, which is in the cytosol. But the other organelles, such as mitochondria, can be used in other stage of cellular respiration. The first stage of cellular respiration, which we call that glycolysis, has 3 stages. The first stage is, or stage 1 is, the investment stage. Why it is called investment stage? Because at this stage, the cells uses 2 molecules of ATP are consumed for each molecule of glucose to be break down. It is called the investment stage because there is the consumption of 2 molecules of ATP in this first stage. Here, glucose is converted into fructose 1,6-bisphosphates in the first stage, and the glucose is trapped inside the cell and at the same time converted an unstable form that can be readily cleaved into 3 carbon units. Here, if you see diagrammatically, in the first stage, 1 molecule of glucose can be converted into fructose 1,6-bisphosphates by passing through the different steps of reaction. We will see how this reaction can be takes place and what kind of enzymes used in the next steps. In stage 2, here is in the stage 1, fructose 1,6-bisphosphate is formed, But in stage 2, this fructose 1,6-bisphosphate is cleaved into 3 carbon units of glyceraldehyde 3-phosphate. That means here is fructose 1,6-bisphosphate. Then at this stage, this compound can be cleaved into 2 compound which contains 3 carbon compound, which is dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. In the stage 3 of glycolysis, it is the harvesting stage. It is the harvesting stage. It is called the harvesting stage because it is the stage at which ATP starts to be synthesized. Here, glyceraldehyde 3-phosphate is oxidized to pyruvate at this stage, and 4 molecules of ATP and the 2 molecules of NADH, which is energy storing compound, are gained from the initial molecule of glucose. That is why it is called as the harvesting stage. It is the stage at which we can get ATP. Here, ATP is a result of the substrate level phosphorylation. Here, at the third stage of glycolysis, the ATP produced is the substrate level phosphorylation. Look, if you see diagrammatically here, glyceraldehyde 3-phosphate, from this glyceraldehyde 3-phosphate, finally it becomes changed into 2 molecules of pyruvate by passing through different stages of chemical reactions, or through passing through a series of chemical reactions. In this case, here at this stage, 1 molecule of ATP is gained, and again at this stage another ATP is gained, and again here totally 4 molecules of ATP gained in the third stage of glycolysis. That is why it is so called as the harvesting stage. Stepwise reactions of glycolysis. Here when you see that reaction 1 means when glucose is converted into glucose 6-phosphate. Now this one is the structure of glucose which is used for the process of cellular respiration or glycolysis. Now in the reaction 1, glucose converted into glucose 6-phosphate. At this time, 1 molecule of ATP consumed, and the process of the reaction can be facilitated by the enzyme which is so called hexokinase. This reaction requires energy, and so it is coupled to the hydrolysis of ATP to ADP. Then the enzyme hexokinase can participate or catalyze this reaction, so glucose gets trapped into the cell or inside to the cell in the reaction 1. In the reaction 2, isomerization can take place. Here, glucose 6-phosphate from the reaction 1 can be changed into fructose 6-phosphate. Now, glucose is changed into fructose in this case. What was done is that isomerization. The process was catalyzed by the phosphoglucoisomerase enzyme facilitate or catalyze these reactions. Then reaction 3 is another kinase reaction. Here that means when the fructose 6-phosphate converted into fructose 1,6-bisphosphate, that is the reaction 3. In this case is another kinase reaction takes place. That means ATP become hydrolyzed, and one of it is phosphate group released from ATP, and it become attached to the fructose. Then it forms the compound which we call that fructose 1,6-phosphate. This 1 and the 6 shows that the position of the phosphate on the carbon atom. And here the reaction is now coupled to hydrolysis of ATP to ADP and an organic phosphate. This is the second irreversible reactions in the glycolysis. Reaction 4, that means when the fructose 1,6-bisphosphate is converted into 3-carbon atom compound, which is dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Both dihydroxyacetone phosphate, it is a 3-carbon compound. Also, glyceraldehyde 3-phosphate is also, it is a 3-carbon compound. But here, you know that fructose has a 6-carbon compound. Now, this 6-carbon compound becomes cleaved into 2 compounds which contains 3-carbon compound in the reaction 4, and it becomes changed into 2 compounds. In this case, fructose 1,6-bisphosphate is splitting into 3 carbon molecules, one which is dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. This process facilitated or catalyzed by the enzyme aldolase. Aldolase enzyme can catalyze these reactions. Reaction 5, here in the reaction 4, 2 3-carbon compound is formed, which is dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. But dihydroxyacetone phosphate is not important for the process of cellular respiration. Therefore, it has to be converted into glyceraldehyde 3-phosphate. Therefore, the dihydroxyacetone phosphate has to be isomerized or to be changed into glyceral 3-phosphate, which is important for the process of cellular respiration. Here, dihydroxyacetone phosphate is not important for the process of cellular respiration, but glyceraldehyde 3-phosphate is important for the process of cellular respiration. Therefore, in the reaction 5, the dihydroxyacetone phosphate has to be isomerized or to be changed into glyceraldehyde 3-phosphate, and the reaction can be catalyzed by the enzyme isomerase. In the reaction 6, this part of glycolytic pathway is so-called the payoff or harvesting stage. It is called the harvesting stage because it is the stage at which the cell starts to gain ATP from the process of chemical reactions. Here, glyceraldehyde 3-phosphate is dehydrogenated by the enzyme glyceraldehyde 3-phosphate dehydrogenase enzyme. Then, in the process, NAD positive is reduced to 2 molecules of NADH, and oxidation is coupled to the phosphorylations of carbon. Then the product is 2 molecules of 1,3-bisphosphate glycerate. Here, the glyceraldehyde 3-phosphate now converted into that of 1,3-bisphosphoglycerate. It is 2 molecules because which is cleaved from the glucose in the 5th reactions. In the 7th reaction, bisphosphoglycerate has mixed anhydride and high energy bond at its carbon 1. At the carbon 1,3-bisphosphoglycerate is mixed anhydride and it has a high energy bond. Therefore, this high energy bond is hydrolyzed by the carboxylic acid, and energy released is used to generate ATP from ADP and inorganic phosphate. And finally, it produces 2 molecules of 3-phosphoglycerate and 2 molecules of ATP. Now, at this stage, that means the reaction 7, 2 molecules of ATP gained. In the process, another compound is formed, which is 2 molecules of 3-phosphoglycerate is produced. Then, in the reaction 8, the phosphate shifts from carbon 3, which is found, the phosphate which is attached on the carbon number 3, can shift to carbon number 2. Therefore, it forms 2-phosphoglycerate. And this process is catalyzed by the enzyme phosphoglyceromutase, can facilitate this reaction. Here, in the reaction 9, it is the stage at which dehydration can takes place. That means the removal of water from the system. Here, the water molecules removed when 2-phosphoglycerate is converted into phosphoenol pyruvate. Therefore, in the reaction 9, dehydration catalyzed by the enzyme enolase or a lyase. Here a water molecule is removed from the phosphoenol pyruvate, which has a double bond between carbon 2 and the carbon 3. That means here this is a carbon 2 and the carbon 3. Between this, because of one molecule of water is removed here, a double bond is formed between carbon 2 and carbon 3. And this process catalyzed by the enzyme which is so-called enolase enzyme. The last steps of reactions of glycolysis, that is the reaction 10. And the reaction 10, phosphoenol pyruvate changed into the pyruvates. Here, in this case, phosphoenol pyruvate is a high energy bond. This is hydrolyzed to form enolic form of pyruvate, with the synthesis of 2 molecules of ATP can be gained by this process. This is irreversible reaction and is catalyzed by the enzyme which is so-called pyruvate kinase, or the pyruvate kinase can catalyze or facilitate this process of reactions. Here, enol pyruvate quickly changed into ketopyruvates, which is far more stable. In the first case, the enol pyruvates produced, but automatically the enol pyruvates become changed into the ketopyruvates, because the ketopyruvate is more stable than that of the enol pyruvate. Therefore, this is to show that the functional group of the keton, therefore it is immediately changed into the keton pyruvate, which is more stable than the enol pyruvates. We have seen that the process of, or a series of reactions that takes place in the glycolysis. Now let me see the glycolysis energy balance sheet. Here, hexokinase in the first stage of breakdown of glucose into glucose 6-phosphate, the cell consume 1 ATP molecule. 1 ATP molecule. Here, negative shows that consumption of 1 ATP molecule. And again, phosphofructokinase again consume 1 ATP molecule. Then when dehydrogenase takes place, 2 molecule of NADH is formed. This NADH is energy storing compound which will help us to release ATP in electron transport chain stage. This is one important energy storing compound which is produced in the glycolysis stage. And the other one is phosphoglycerate kinase. At this stage, again, 2 molecule of ATP is gained. Then in the pyruvate kinase, 2 molecule of ATP is also gained. Now, from the total, how many ATP is gained in process of glycolysis? It is 2 plus 2, which is 4 ATP is gained. But in the process, the cell consume 2 molecule of ATP, minus 1 and minus 1, which is 2 ATP. Therefore, if you subtract 4 minus 2, a net ATP gained by the process of glycolysis is so-called 2 ATP. And also 2 NADH, which is high energy storing compound. And again, 2 molecule of pyruvate is formed at the end of glycolysis from 1 molecule of glucose. The 6 carbon atom compound glucose is converted into a 3 carbon atom compound here in the pyruvate. This shows that this is not completely oxidized at this stage. Therefore, the cell needs to completely oxidize this 3 carbon compound which is pyruvate. So, to do that, what will happen to this pyruvate? So, what will be the fate of the pyruvate? We will see after this slide. Here, to summarize the process of glycolysis pathway, here in the reaction 1, phosphorylation of glucose by ATP, that is here the reaction 1, here reaction 2 and 3, In this case, rearrangement followed by the second ATP phosphorylation, and reaction 4 and 5 at this step. It is the 6 carbon molecule is split into 3 carbon molecule. That means this is a 6 carbon molecule and split into 3 carbon molecule. And reaction 6, oxidation followed by phosphorylation. Reaction 7, removal of high energy phosphate by 2 ADP molecule to produce 2 ATP. Here is 2 ATPs generated. And reaction 8 to 9, in this case it is removal of water, which yields phosphoenol pyruvate. Here the water molecules is removed, which is facilitated by enols, and phosphoenol pyruvate is produced in this case. Then reaction test, the removal of high energy phosphate by 2 ADP. As a result, 2 molecule of ATPs gained. This is this summary of the glycolysis pathway. But at the end of glycolysis, 2 molecule of pyruvate is obtained. But what is the fate of this pyruvate? In the net reactions of the glycolysis, 2 pyruvate is formed, and again 2 ATP is formed, 2 NADH is formed. But what is the fate of this pyruvate? What's the fate of this pyruvate? Here, the pyruvate which is produced at the first stage of cellular respiration, which is glycolysis, it can be changed into 3 points, or it can be undergoes 3 processes. But this process depends upon the presence or the absence of oxygen. And the presence or absence of oxygen determines the fate of this pyruvate. Here, when there is plenty of oxygen, pyruvate is first converted into acetyl-CoA if there is plenty of oxygen. However, in the absence of oxygen, if there is no oxygen, in vertebrates, pyruvate is converted into lactate or lactic acid. Whereas in yeast or other microorganisms, the pyruvate can be converted into ethanol and carbon dioxide. You know that ethanol is alcohol. Now, let me see this diagrammatically. The fate of pyruvates. Here, the fate of pyruvate is determined by the availability of oxygen. If there is plenty of oxygen, the aerobic organisms can convert, or in the cell of aerobic organisms, this pyruvate can be converted into acetyl-CoA. Now, this acetyl-CoA can enter into the second stage of cellular respiration. But if there is no enough oxygen, and in anaerobic condition, it can be converted into lactates by vertebrates. If there is no oxygen. But in the microorganisms, examples such as yeast, it can be converted into ethanol and carbon dioxide. This is the fate of pyruvate, which is produced by the process of glycolysis.
Thank you for joining me today. If you enjoyed this lesson, hit the like button, subscribe, and share with your friends.